afternoon, Dr. Khandare. Such a pleasure to have you over here. Thank you so much for coming. And also, we are all attending the fantastic Bonds 2025 meeting, which is the first time hosted by 1Cell.ai in Mumbai at the Westin. We're really enjoying it. A lot of medical oncologists, academicians, investors, VCs, they have to be at here. But I wanted to take a few minutes to speak with you. Say thank you for coming. Dr. Jayant Khandare is actually a scientific officer and co-founder, one of the co-founders of 1Cell.ai. We are here to just interview with him to understand what his journey has been from the time of when he decided to study to science to how he's taken us to where we are. So he really appreciate all the previous meetings. So this question I would have for you is to ask you if you could share a little bit about your journey from how we got into science and technology from you.
Thank you, Dr. Malini. It's a privilege to talk to you. Pleasure always, interacting with you. So my name is Jayant and I'm a Chief Scientific Officer of 1Cell Diagnostics. I'm a chemical engineer by PhD from National Chemical Laboratory. And I worked in US for several years, 4 years, at multiple institutes with a background in biology, cell biology, drug delivery systems. And always wanted to return to my country, India, who actually sponsored my all education from standard 1 till my PhD with no cost. So I feel that I have an obligation to revert to my society, especially Indians, and I feel extremely patriotic about transforming that science translations through my education to Indians. That's my journey about what I want to do and why I'm actually in India and why I'm with 1Cell.
I do know that outside of your stay in US, you were also in Germany, in Berlin.
Yes.
You share.
So I returned to India and worked in one discovery company, and I realized that I need to still have more education. So you and me, we became Humboldt Fellow, Alexander von Humboldt. I think that's a common thread that we would always remember. And I went to work as an Humboldt Fellow in Berlin, in Free University, working on polymers and material science. Actually, all these experiences requires, good knowledge, good understanding, good updation. And Germany gave me that opportunity to work on material science. That actually later on translated into all my technologies.
That's really very important, and that's why I asked about specifically about that. The question of the stay in your work in Berlin, because that then leads to my next question, which is, how did you didn't, the word transport is not really doing justice because he didn't transport it. He very deliberately thought about it. And so he used the fundamental knowledge that he had in the majority sciences progress. So if you could briefly give us that, how that led to the whole year was.
In 2010, as an head of an innovation in one of a drug discovery company here in Mumbai, I tried to sense what are the most challenging aspects of my science. And I stumbled upon a paper in Science which talked about cancer circulating problem. But in order to look at a tumor cells which are in a circulation 1 in billion with their ability to survive for a long time, and as their abundance is extremely low, the chemistry and the chemical engineering could actually design materials which could capture these rare cells, which becomes very expensive, exorbitantly expensive technology in US. So I challenge myself that as a chemist or as an engineer, what is the problem I wish to solve? And capturing circulating tumor cells was number 1 amongst top 10 science problems in 2010. So that's how actually I stumbled incidentally on CTCs. And then I started my entrepreneurship through Actorius. We got India's first license as a medical device in 2019, ever designed and clinically validated in India, in our patients with Tata Memorial Hospital, with Dr. Pankaj Chaturvedi, with Dr. Kumar Prabhash. So I feel proud that science translation happened in the extreme of a challenging problem that actually I was looking at it. So it took 8 years for me to make India's first technology. And then the second journey started for innovation with 1Cell Diagnostics.
So that was fantastic. So congratulations for that.
Thank you.
We didn't stumble upon it. You purposely but we used your this foundational knowledge in the material science and just translating it into the biological arena. So as you know, for all of you to know that he at one of the who is key initial pioneer, need isolating CTCs, CTCs and single cells of TPCs. So circulating tumor cells, which is a whole different concept. Then there's gold standard tissue biopsy, the TPG, and then liquid biopsy, which is primarily ctDNA. Now moving on to what is coined as cell biopsy, which is a term that is being coined by men and the other co-found base. In more importantly, looking at circulating tumor cells because they capture what's happening live. They are live and do it as dynamic, unlike tissue that expects and pulls up in terms of what's happening with the patient's tumor. So that being that when you discover, you found TTC and you discover that you could isolate single cells, all that. And just a little bit about how you felt. Did you have the aha moment? Did you feel excited? Were you nervous? How did it?
See, before capturing those rare cells, right, from the blood of a cancer patient, I think most important aspect that happened when I met Dr. Gowhar in one of a meeting in Delhi. So I think destiny brings, good people together to solve larger problems. So meeting Dr. Gowhar, then Mohan in about 2019 and 2020. So I realized that beyond enumeration of a circulating tumor cells, which is now its standalone technology, and we have done now almost 9,000 patients in India centric and other countries. But when we look at a generation 2 circulating tumor cells, we wanted to design new materials because you want to capture one cell at a time, which would have ocean of an information from the genomic profiling. That's what Dr. Gowhar and information team could do it. But I had to redesign chemistry and engineer those materials, which was uh, comparatively very easy than my earlier technology. So when Mohan mentioned about capturing live cell, we could tune a chemistry which could capture a cell one at a time from the blood sample, and we could release them without contamination of a leukocyte. So I think crux of creating this cell biopsy terminology that what Dr. Gowhar later on coined is an extraordinary mission impossible, as I mentioned today in my talk. But because we were interdisciplinary team, we could isolate cells on now 1600, 1700 patient samples. We have a CTC cell biopsy bank with a live cells as well as DNA with the libraries. I don't think so anyone has looked at a protein as much as we are capable today at single cell. So, in sum up, I think single cell live capturing happened only when I met Dr. Gowhar and Mohan and formed one cell, because one cell is also important than other cells. And I hope today's this polls conference make it realistic and worthy to the name of a company.
Of course. And so that's been actually, that's how the journey evolved. Mohan CA, Gowhar Shafi, our chief geneticist, bioinformatician, our CSO, and also with Ivan Hay, is a guest baby with talking to. So now the report here upon this company, the goal is to reach 1 million patients at patients in India, and then also in a global outreach with this affordable, actionable test, diagnostic test. What do you foresee? Because you are such a visionary and you have been, you're the one because of whom this company exists with the CGCs. What are you thinking? I'm sure your brain is always thinking, but some new thing would happen. What do you think the next 5 to 10 years one cell AI needs to do to be the global leader? anybody and everybody would think about only one cell as a company would go to.
Although we are talking about a one cell at a time, so I think all co-founders names that you mentioned, I think we bond each other. There is a bond in our technology that there are multiple bonds. I think four of us bring different capabilities on the board. So uh, that covalent and non-covalent bond is actually between co-founders, which we have kept it intact for 3 years. So vision is extremely challenging because our goals are very extremely of an importance to cancer patient reaching 1 million patient. I hope we meet to that goals and challenges. The field is actually getting into the precision oncology with AI powered system as strong we are actually, and we are able to detect cells. We can use AI system to predict the genomic profiling, but we could also take bright field images, apply algorithms, and account the cell type using only a black and white image. So I feel that OneCell will be at the forefront of single cell technology. Today we are from the cell biopsy bank and information with the clinical trials in US and India. I think in the next 2 years of a time, world will realize there has to be a new way of discovering drugs over to the traditional drugs after spending billions of dollars. So our major contribution will will be in the science translations through innovation powered by what we do, combiningly with all other breakthroughs, is contribution in actually a next generation drug discovery. And our multiple clinical trials with KOLs is actually generating that data as a proof of principle.
So that takes it to a whole new. It's not just next generation, which multiomics, a next generation cell biopsy, but next generation drug discovery, drug development, and so obviously clinical trials and how we recruit for clinical trials that also needs to undergo a significant, rapid way to be able to select and kind of that needs to evolve to do not think that.
I think discovering a drug is extremely challenging, as you have seen in US. But if we could reduce this cycle of time, of validations, of patient inductions, when you don't have a tissue, there are no alternatives, and that delays whole of a process. And in the end, we don't achieve a targetability either on the cell surface or in the nucleus or cytoplasm. So having a live cell actually could be capable of this multiomics and transcriptomics. So I think we will be considered as a pioneer. I hope so. I'm very optimistic in this, that this will lead to a new level of a science of a drug discovery, which will not only enhance the targetability, but it will reduce tremendously the process time that goes in a discovery.
So this everything seemed very, very exciting. I think we're waiting for your next innovations. Your next, not just innovation, it's going to take it from good to great. And that's where we want to be. I think we've seen everything evolve, and your mind is constantly on the job, on the go. I'm sure every waking moment, every sleeping moment. So we are just very excited to see what's next. And also, I think for OneCell.ai, we want to see how we think will it be, as I said, the household thing, wherever you go. Of course, we don't want the world to be ridden with cancer patients, but whichever household has got cancer patients, being a disease that's really taken a lot of the world, we want to see how we can be there in people's minds, in patients' minds, in physicians' minds, in pharmaceutical companies' minds, and last but not least, the payers. So in countries where it's not self-paid, we want to make sure that we are thinking about what good we are doing, be able to handle this. So thank you so much for your time today,
My pleasure.
Dave, and we really look forward to the next checks. Leap.
Thank you very much.